Okay, hi everyone, and welcome to the fourth lecture of Biomaterials 1. We're going to continue our conversation about polymers by discussing naturally derived polymers and also hydrogels. Hydrogels can be prepared from natural polymers and synthetic polymers, but I just thought, so that's why I thought I'd introduce natural polymers first, and then we can discuss hydrogels in the context of you can pretty much make one out of both types of polymers. Okay, so what is a natural polymer? A polymer, if you remember, a polymer is just a long chain of repeating units, and so that's also what a natural polymer is, but it exists in nature. So DNA is considered a polymer, it's a polynucleotide, but really when we're talking about natural polymers, it's generally either proteins or polysaccharides like proteoglycans or glycosaminoglycans. So the common proteins are silk, that's a really popular biomaterial that's being used now, and collagen, that's probably the most common naturally derived biomaterial. But there's a lot of different kinds. These are just some. Like I said, collagen is the most widely used. It's found in connective tissues naturally in mammals and other animals, but in humans as well. So you find a lot of different types of collagen in the body, and its main function is mechanical, and it's also, it is important in swelling and the water properties of tissues, but it's mainly mechanical, and it also does have some properties in letting cells transmit signals from the extracellular matrix, and I guess what I'm thinking here is mechanotransduction. Elastin is a common one when you're trying to mimic an elastic tissue like ear cartilage or a tendon or ligament. Fibrinogen is a really popular one for making hydrogels because you can make a fibrin hydrogel out of thrombin just by combining thrombin and fibrinogen solutions, and that's involved in blood clotting. So those are your major proteins. As far as polysaccharides, that's just repeating units of sugars, and so alginate and chitin and glycosaminoglycans are examples of polysaccharides. And we'll learn more about alginate in the labs for this class. But alginate is actually derived from seaweed and algae, and chitin is derived from the protective shell around insects and crustaceans. And so the chitin is definitely used for shape and form and mechanical properties. Algae use alginate for energy storage, but we just use it as just a hydrogel, just to make hydrogels that have various applications, which we'll get into later. Glycosaminoglycans are found in connective tissues, especially cartilage, so they're a really important part of the extracellular matrix in these tissues, and they're very important for resisting forces, either in tension or compression. And then you have your polynucleotides like DNA and RNA, which are mainly used for protein synthesis by the cell. But you can use all of these polymers directly for biomedical applications, or you can incorporate them into materials that have other materials, such as synthetic polymers, and that would confer biological function, because these materials are biomimetic and because they're found physiologically. So it's a way to add bioactivity or biofunctionality to a synthetic material. Okay, so the advantages of natural polymers and using that as a biomaterial is because they're natural, they're biomimetic. So they have physiological, mechanical properties in theory, but because their organization in tissues is really specific, we actually can't fully mimic the mechanical properties of tissues, and that's an active area of biomaterials and tissue engineering research, is trying to actually make, for example, a hydrogel that has the same mechanical properties as cartilage, for example. But they are bioactive, so that means that cells have receptors, or they can interact with with sites in the proteins or in these natural polymers. So that's nice, but on the flip side, that makes it more complicated to manipulate them. And because they're being synthesized in the body, we're not synthesizing them, they're more complicated to manipulate. And for the same reason, you have natural variability from different sources, and especially interspecies variability, but just batch-to-batch availability. So if you buy collagen that was isolated from, bovine tissue, it will be different every time you buy it. So that's something to consider. A nice advantage is that it's degraded by naturally occurring enzymes and typically metabolized into safe and natural byproducts. And that's nice because for a lot of goals in biomaterials and tissue engineering are trying to grow, or trying to get the body to regenerate tissue that it otherwise would not. So that's nice that it could be slowly replaced by healthy and regrowing tissue. But that means that if the mechanical or if the degradation profile is not what you would want it to be, you're going to have to physically modify that or chemically modify it in order to make it a better degradation profile. And then that will, of course, affect its mechanical properties and its bioactive properties. So just by changing the degradation properties, you could get rid of all the reasons why you decided to use a natural polymer. Because they're biomimetic, you have less of an inflammatory response, sometimes, is my qualifying statement there. And of course, that depends on the polymers that you're talking about. But because the body doesn't think, or I guess not to anthropomorphize, the inflammatory response, but it doesn't think it's as much as a foreign body as, say, a synthetic polymer, because it is similar to the tissues to what you would see in the normal body. So you might have less of an inflammatory reaction initially, but a lot of times you'll have an immune response from the adaptive arm of the immune system that, when you develop antibodies to this non-self material that you're putting in, especially if it comes from a different species, which it generally does. And there's also the possibility for disease transmission, since it does come from different species, but it's close enough that our body can recognize it. So that's definitely to think about. But the nice thing about, in conclusion, for the advantages of natural polymers, they're just like synthetic polymers with all the properties that we talked about last week in terms of viscoelasticity and everything, but they have the possibility for bioactivity. So that's useful. Okay, so the first group of natural polymers that I want to talk about is proteins and peptides. And I'm mainly going to talk about these in terms, or in the context of collagen, because it is one of the most widely used natural polymers. So like I said, it's found in skin, tendon, cartilage, and bone. They just keep discovering different types of collagen that exist physiologically. So right now there's at least 15 different types. And they're degraded by collagenase and by other matrix metalloproteases. All collagens share the triple helix configuration, which is important for its structure. And their structure is really important for their properties, from the primary level to the quaternary level. So I think it's helpful to know, to start from there. So the primary structure is the amino acid sequence, and that's actually that's actually the repeating units of the polymer. And so, of course, that's important, and they all have their own functional groups that come off, and they all have their own, all the amino acids have their own different ways that you can modify them chemically just through simple chemical reactions. The secondary structure is the chain configuration, which is generally determined by bond flexibility. So just like we talked about with normal polymers, whether or not a bond can easily rotate determines their mechanical properties and a lot of their properties. So that applies to natural polymers like proteins as well. And then the tertiary structure and the quaternary structure are extremely important for protein bioactivity. And so that's something that's a little bit different from synthetic polymers. For bioactivity of collagen, they need to be in a specific conformation. So the tertiary structure is sort of just like how the chains pack together, but the quaternary structure is supramolecular, so it's how different molecules of collagen interact. And that's really important for protein receptor recognition and for basically so cells can interact with the proteins. So the quaternary structure is what you actually need to retain if you want there to be bioactivity. The cells might still be able to react with the polymer, but if the protein doesn't have that quaternary structure, then it's not, it won't have the physiological effects that it would natively in the body. Types of collagen, there's more than 15. They all have different functions, but they're mainly mechanical and involved in transmitting signals between cells and the extracellular matrix. In my doctoral research, I worked really closely with cartilage, and so, for example, collagen 2 is a huge component of cartilage, and it swells with water, and so but it's also somewhat cross-linked in cartilage, so it's responsible for resisting the changes in swelling, or resisting the forces and the swelling that it's responsible for resisting the forces, which is also aided by swelling, and that's why cartilage can withstand such high loads in compression, even though it's mostly just protein and water. It's mostly water. Okay, but there's other different types of collagen, and they're all together in fibrils. They all pack together in a triple helix, and then that's packed together into fibrils, and that's why it's really strong in tension for a lot of the collagens, like the collagen that's involved in tendons. Okay. So collagen-based biomaterials are extremely diverse. They're used as sutures, as a tape, and that's why the tensile properties are really important. They're used as hemostatic agents to promote blood clotting and stop bleeding, and so then you can have collagen in a powder or sponge. They've also been used to make tissue-engineered blood vessels, or just to replace blood vessels, which I guess is also tissue engineering, depends how you define it. And so it's interesting that, so a lot of these say that the physical state of the collagen for these applications are processed blood vessel or processed porcine tissue. So a really big area of biomaterials is actually taking animal tissues and decellularizing them to remove anything that might cause a adverse immune reaction, and then using it for a different application. So, or for the same application that it was. So porcine is the most common, I guess, because their genome is pretty homologous to humans, and so I guess their tissues are like the closest to humans that we can get, and so for heart valves and tendons and ligaments, you have these decellularized tissues that go through all these washing steps, and they just implant it right back into the heart valve to have a replacement heart valve, and it's used pretty successfully, but they're still undergoing optimization and research. And then, let's see, the burn treatment is a pretty good application of collagen. So a porous collagen sponge will be hydrated in saline and then placed over a wound, which would aid in the healing process. And of course, it's really useful in tissue engineering as well, because cells really attach to collagen and like to produce tissue on collagen. But you'll learn more about that in the tissue engineering sequence. Okay, so like I said, the quaternary structure is really important for all the properties of collagen, for the bioactivity of its, it's the specificity of its bioactivity. But you can also use collagen if you remove the quaternary structure. It just has different properties. So if you actually heat it above 37 degrees, it turns into gelatin, or what we know as gelatin. And this can actually happen in the body as well, because it's only at 37 degrees. And that's when collagen will be degraded. It's originally turned into that randomly coiled structure that I guess could be called gelatin, and then it's more easily degraded by MMPs. So gelatin is really common in tissue engineering and also in drug delivery, so you'll learn more about that in the next sequence of this course and the third. And it's much more rapidly degraded than collagen, so that's how if you wanted to control the degradation of a collagen sponge or a collagen scaffold, this might be one way that you do it. But when another way that you can physically modify the structure is that banding that's characteristic of collagen. So that's a picture of collagen that has that characteristic banding, and that means that the quaternary structure is intact. If you lower the pH below around 4.25, that banding will disappear, but the tertiary structure of collagen is still there. And so that is how researchers discovered that it's the quaternary structure of collagen that's really important for hemostasis and clot formation. So when they found out that the quaternary structure is important for clot formation, if you wanted to make something that does not form clots, so you want it to be less thrombogenic, by lowering the pH to get rid of the banding, that's how you would do that. The next most common way that collagen is processed for biomaterials is a porous collagen sponge. And so a really common way to do that is to make a dilute collagen solution, just an aqueous solution of the collagen protein, and then freezing it and freeze drying it, so lyophilizing it under low temperature vacuum. And the ice crystals form because it's an aqueous solution, and then when they sublime, they leave pores behind. And so you can actually control the pore size and orientation based on, well, so if you lower the freezing temperature, you get smaller pores. And so because you'll get smaller ice crystals, and then when they sublime, you have smaller pores. So that can be varied between, I think it was like 1 and 800 microns. So that's actually a really big difference. And you can actually have directional pores. You can have channels or directional pores by controlling the direction of the freezing. So if you have it freezing from one side and going up or in one direction, that's the direction that you'll have pores aligned. And so that could be really important in tissue engineering applications. So that's good to know. And the interesting thing about these collagen sponges, after you freeze dry them, is they retain their quaternary structure. So they're intrinsically hemostatic. So that's why they're pretty good for wound healing, because they'll stop bleeding that way. Okay. Then a lot of biomaterials engineers will cross-link the collagen matrix, which will really affect its degradation profile. So if you cross-link a material chemically, especially a natural polymer, it can no longer be naturally degraded by the body because those cross-links are chemical, and the cells or the enzymes won't cleave in the places that they used to. So that's really useful for some applications. For others, it's not. I mean, if you don't want it to, if you need it to degrade, of course, the cross-linking would not be a good thing, and it is permanent. So there's that. But this is an example of the Integra Life Sciences bilayer wound matrix is an example of a cross-linked collagen matrix that's actually used as sort of like a skin graft or something that's used to encourage skin regeneration for burns and other dermal wounds. Ossigen is another cross-linked collagen matrix. And that is actually minerals like hydroxyapatite dispersed in collagen fibers, and so it's biomimetic of bone. It doesn't have the same structure, which is why it doesn't have the same mechanical properties, but it is somewhat osteoinductive in that it might help to cause bone ingrowth in the defect. But so the indication for these sponges is that you would take a bone marrow aspirate in the surgery room and use the sponge to suck up the bone marrow aspirate, which has stem cells and other good healing things in it, and then stick that in the bone defect and it heals better. So that's a pretty good use of collagen matrix. Okay, so those are some physical modifications of collagen. You can also chemically modify collagen. So I briefly mentioned that you can chemically cross-link them, which will decrease the rate of degradation or not let them be, they won't be degraded at all. It may reduce the immunogenicity, but that is only if you cross-link the antigenic sites, the sites that your body would produce antibodies in response to. So there's no guarantee that just simply cross-linking with a non-specific or cross-linker like glutaraldehyde or other aldehydes would reduce the immunogenicity. But if you targeted those antigenic sites on purpose, then you would. The problem with cross-linking is that any residual glutaraldehyde or other aldehyde is cytotoxic precisely because it cross-links proteins. So glutaraldehyde works by just having a chemical reaction with free amine groups. So the aldehyde just chemically binds to the amine groups. And so if you have a dialdehyde with two aldehyde groups on either end, it acts as an actual physical linker. So if any of that gets released to cells, then that would cross-link proteins in the cell membrane and kill them, because you need fluidity in your cell membrane. And also there's some research that has shown that the inflammatory response to cross-linked materials is much, much worse than if you don't have cross-linking. And that's probably because it's less natural. You know, you don't have that chemical cross-linking in the body, so you just sort of took away the whole reason why you use natural polymers and that they're biomimetic. But you know, I think that depends on the degree of cross-linking, and it could be a really useful tool for modifying the properties of collagen sponges. Okay, so I talked a little bit about xenografts. So that's when you take a tissue from one species and implant it in the other. So of course we're talking about implanting into humans because these are medical problems. And so another example of a xenograft is decellularized porcine small intestine submucosa, or SIS. And that is basically you take some of the lining of the small intestine in a pig and you decellularize it, like I said earlier, and then it's actually used to repair soft tissues and maybe like abdominal wall reconstruction. So it's used internally and also for stronger tissues like tendons. And so it works reasonably well, but you have the same issues of sometimes it needs to be cross-linked in order to make the properties stronger and have longer stability in the body, and then you might have more of an inflammatory response to that. But we are going to talk about that more in the lecture about the foreign body response. But you also have religious and cultural issues, because there's a lot of religions and cultures that don't want other animals implanted into their body. So that's definitely an issue for trying to make something that would be translatable to humankind. There's huge populations that, of course, wouldn't want pig or cow in them. Okay, so that was mostly collagen that we talked about. There's a lot of other common natural polymers like silk, hyaluronan, fibrinogen, phytosan, agarose, alginate, and they're derived from multiple sources, but a lot of these are used to make hydrogels. So I'm going to go into hydrogels now, and then we can come back to more specific examples of when you would use these polymers. Okay, so hydrogels are 3D cross-linked networks of hydrophilic polymers so that they swell with water. But they're cross-linked so that they have elastic responses, and also so that they're not soluble in water. So even though they're hydrophilic polymers and they start from a solution of polymer in water, when they're cross-linked, they're not soluble. So you can see it and feel it, and it's got a 3D structure, so that's how you know that it's hydrogel. So this is a picture of a really cool hydrogel that was just published in Nature like last week. I encourage you to go check out that video. This is just a screenshot of the video where the hydrogel is super strong, but it can deform up to like 1,000 times its original shape or something, so it's super strong and super stretchy, and it's pretty cool. And then the other pictures are examples of using hydrogels for cartilage replacement. So if you have an area of cartilage that is damaged, it's been proposed that you could replace it with a synthetic hydrogel, or even a hydrogel made from natural polymers, which is still man-made. And I'm going to talk about that in a little bit more detail later in this lecture, so I will wait on that. Okay, so like I said, hydrogels are cross-linked networks of hydrophilic polymers that are highly swollen with water. They can be made from natural or synthetic polymers. They can be physically or chemically cross-linked, and they can be environmentally responsive. So there's a lot of different ways to categorize hydrogels, and a lot of different properties that you need to consider when you're talking about hydrogels. They can swell with I set up to 95% water, but that number is always thrown around with different, I've seen 99% water and 90% water, so it's just a lot. So the nicest thing about hydrogels is that they have similar material properties to tissues, or at least more similar than other materials we have, like just dry polymers or ceramics or something. well, actually ceramics are biomimetics, so more than metal. So it's mostly because their water content is so high, just like tissues, that we see that the corresponding properties. So in terms of the mechanical properties, their lubricating properties, and transport of nutrients and oxygen through hydrogels, they're all really similar to tissues, or at least you could modify the hydrogel to make it really similar to tissues. So hydrogels are really useful in drug delivery because you can control release of drug or protein based on diffusion, which you can control based on properties of the hydrogel network. And they're also really useful in tissue engineering because encapsulated cells interact with the hydrogels in a similar way to their native extracellular matrix, depending on the type of polymer that is used to make the hydrogel, of course. But it also allows, hydrogel encapsulation of cells also allows nutrient exchange because diffusion is really good through hydrogels. So because of the high diffusion of gases and liquids through hydrogels, it was one of its first uses was for contact lenses because the problem with contact lenses when they were first invented in the 50s was that oxygen could not diffuse through the glass that they were using to make contact lenses. So that didn't really work. But when they switched, and then I was also, I think you guys saw in the first lecture, which was about the history of biomaterials, there was also a generation of contact lenses that were just PMMA plastic, which was also not really permeable with oxygen, so they had holes in it to allow oxygen to go through, and that didn't really work because there still wasn't sufficient oxygen diffusion. But when hydrogels were invented in about the 70s, and they had excellent oxygen diffusion, that really revolutionized contact lenses, and now they're widely used. Okay, so hydrogels can be classified in a number of different ways, and you'll read, there's a lot of review articles out there on hydrogels, and you'll read a lot of different ways that they're classified. I've seen them classified based on the nature of their side groups, so they're classified as either ionic or neutral. I've also seen them classified based on their network morphology, and this is a lot more kind of confusing, so they can be amorphous, semicrystalline, hydrogen bonded, based on some supermolecular structure like collagen. And then I've also seen them based on their porous structure. Microporous, macroporous, or non-porous. That's probably the most confusing because there's no such thing as a non-porous hydrogel. It just means they have very small pores. So I don't like any of these classifications. The way that I like to classify hydrogels is if they're physical or if they're chemical/covalent. So physical hydrogels they're still 3D cross-linked networks, but the cross-links are not permanent. So even the hydrogel might stay permanently as a hydrogel, but the actual cross-links are not chemical, they're not covalent. So they're either hydrophobic interactions, hydrogen bonds, or ionic forces. And if it's a chemical or a covalent hydrogel, then that's actually permanent cross-links that are chemically bonded, covalent bonds between different polymer chains. So every single hydrogel can be classified as either physical or chemical, so that's why I think that is a good method of classification. Okay, so we can talk about physical gelation in a number of ways. So there's hydrophobic interactions, and so that's when you might take a hydrophobic polymer and make it more hydrophilic by changing the side groups to be more polar and more hydrophilic, by hydrolysis or oxidation. So then you have a polymer that is, maybe it has a polymer, a polar backbone or a non-polar backbone because it's a carbon backbone, but then it has polar side groups. And so the non-polar backbone wants to be together because of hydrophobic interactions, and it puts the polar side groups towards the liquid because it's in an aqueous solvent. So we're always talking about these polymers dissolved in water. So just that interaction between the hydrophobic groups within polymers is actually what causes the insolubility of the polymers in the water. But since the polar groups are still swollen in the water, it becomes a solid. So it's actually an insoluble polymer that is swollen with water. So that's physical gelation by hydrophobic interactions. You can also have ionic gelation, where you have ions that are holding together a charged polymer. So you're going to work with calcium alginate in the labs, and so alginate is a polyanion, and when you add a divalent cation like calcium, you have the cations hold together the different parts of the polymer because of ionic interactions and just electrostatic interactions. So that's a pretty common one. These hydrogels are interesting because you have to assume that the ions are going to exchange with the ions in the body. So if this is something that you're going to implant, the ion composition might change, which would change the properties of the hydrogel. So that's why it's really important to understand the cross-linking of ionic hydrogels. And then if you're going to study these hydrogels in vitro, so in lab experiments, this hydrogel might dissolve in something like PBS that only has monovalent ions. You need to have the divalent ions to keep it together. So that's just something to think about. Okay, so those are your primarily your physical hydrogels. I didn't put a diagram or a schematic of the third most common type of physical hydrogel, which is hydrogen bonds. And so, or hydrogen bonds hold together the chains. I'm surprised that I forgot to do that because my research was really heavy on PVA hydrogels, polyvinyl alcohol hydrogels. So that was what my doctoral research was on. And that is a long carbon, polyvinyl alcohol is a long carbon chain with hydroxyl groups coming off of it. And so what happens with aqueous solutions of polyvinyl alcohol is that you can actually freeze them, and the ice crystals cause the chains to align and cause crystallites to form in the polymers, and then when you thaw it, hydrogen bonds form between the hydroxyl groups on adjacent chains. And so the more you freeze it and thaw it, the more alignment you have and the more formation of crystallites and aggregates, and the stronger it gets with each freeze-thaw cycle. So the most famous example of a physically cross-linked hydrogel that's cross-linked by hydrogen bonding is PVA hydrogels. Okay, so those are your physical hydrogels. We also have chemically cross-linked hydrogels. So there's a couple different ways to do it. They all result in just covalent bonds between polymers, and they're swollen with water. So this schematic is showing that you might have a bifunctional monomer or polymer, and then when you put in cross-linkers, you have the monomers, they form a polymer, but then there's the presence of water, and the multifunctional cross-linkers connect different chains, and that forms a hydrogel network. You can actually even do this not in the presence of water, and then you could swell it later, and it's still considered a hydrogel. You could also have polyfunctional polymers with where their pendant side chains are reactive, and then they form a crosslink. You don't need a crosslinker to actually connect them if they have their own long enough side chain that could connect to another one, and then that would form a network. Other ways are that you might polymerize a monomer in the presence of a crosslinker into a polymer, and because there's also a cross-linking agent there, that would cross-link different polymers together, forming a 3D network, which is a hydrogel once it swells. Or you might have longer monomers that are referred to as macromers, and it's the same concept there. Or you might have full-on water-soluble polymers, and then you add a crosslinker there, just like a shorter chain one that actually forms the crosslinks with the actual crosslinks, and that's a hydrogel network. Interpenetrating hydrogel networks are kind of interesting. Those are abbreviated to IPN networks, or IPN hydrogels, interpenetrating network. So you might have heard about them. And that's when you have a hydrogel network and you add in the monomers to a different polymer in the hydrogel network, and then you cross-link them, and they form a network all dispersed around the hydrogel that you started with. So you have two hydrogel networks that are all interpenetrating, so that's why it's called that. And that's cool because the properties that they have is like a composite between the two different hydrogel networks. So that might be a really interesting way to take two advantages of two different materials and combine them. Okay. So that's how you make hydrogels and how you classify them. There's properties of the network, of the hydrogel network, is what is going to determine its material properties, including its mechanical properties, its structure, swelling behavior, and diffusion, which is going to be really important for drug delivery. So we'll talk a lot more about that in biomaterials too, and really important in tissue engineering. If you're going to encapsulate cells, there needs to be diffusion of oxygen and nutrients. So that's really important in tissue engineering, so we'll talk about that in biomaterials 3. But for now, I want to talk about porosity. So I was saying that I don't like this as a classification because non-porous hydrogels are still porous. They just have pore sizes that are between 1 and 10 nanometers. Microporous, I really don't like that name because they're not micro-sized pores, they're nano-sized, they're between 10 and 100 nanometers. Same reason why I don't like macroporous, because they are micro-sized. And for my doctoral research, I actually worked on superporous hydrogels, which is sort of like not really an official classification, but it's something that we used the term for. And so we made very large pores in the hydrogels. I think that scale bar is 500 microns. Oh no, it must be 100 microns because then I have a graph of the pore size there. And the pore size ranged from 40 to 100 microns depending on the processing parameters. And that can be useful if you want. I wanted to have cells from the surrounding tissue grow into the hydrogel in order to have some integration between the surrounding tissue and the hydrogel, so that's why I wanted large pores. So that's basically the porosity. So when you're thinking about the pores of a hydrogel, so superporous is kind of obvious why we call it that, because they're really big pores, but they're sort of different from the other porosity classifications that I have up here, because within those superporous hydrogels, the walls of the pores are made of hydrogel that itself could be classified as non-porous, microporous, or macroporous. So the reason why is that pore size is actually the mesh size of the network. So if you look at the literal distance between chains in the hydrogel, between polymer chains, so that actual distance is the mesh size, also known as the pore size. So if we go back to the picture of my superporous hydrogels, so because the hydrogel walls had they were networked themselves, they had their own corresponding mesh size within their network. Okay, but so the mesh size and the cross-linking density determine almost all properties of hydrogels, so they're really important. Kind of intuitive to understand. The mesh size is just the distance between cross-links. So that's kind of like porosity. So you can see how that would affect properties, especially swelling properties, because if that pore size is bigger, then it can accept more water into the hydrogel network, and then it'll swell a lot more. Cross-linking density is very related to mesh size, and that is the distance. Well, cross-linking. The molecular weight between crosslinks, so MC in this diagram, is the actual distance between crosslinks. So if you had multiple polymer chains cross-linked, the average distance between two links is the molecular weight between crosslinks. And you can calculate the degree of cross-linking by dividing the molecular weight of the polymer's repeating units over the molecular weight between crosslinks times 2, which is derived from rubber elasticity theory, and so I'm not going to get into why there's a 2 there. And then, so degree of cross-linking is one parameter that you could use to describe hydrogels, and then another is cross-linking density, which is extremely related, and that is just 1 over the specific volume of the polymer times the molecular weight between cross-links. And the specific volume of the polymer is just the inverse of the density of the polymer, and it's in amorphous state. So the cross-linking density is really important because that's going to determine a lot of their properties. So in what way does it determine their properties? Like I was saying, if you increase the pore size, which means you decrease the cross-linking density, you are going to have if you decrease the cross-linking density, you have an increase in gel swelling because more water can go in there. If you increase the cross-linking density, so you make the mesh size smaller and the molecular weight between cross-links smaller, so that the cross-linking density is increased, then you have an increase in mechanical properties like modulus or stiffness, and that's because they can withstand more forces because they they can't move as much in response to an applied force. So that's why it's stiffer there. So because the cross-linking density is so important, I want to go through the derivation of how you can calculate the molecular weight between cross-links so that you can then in turn calculate the cross-linking density from swelling experiments. Because you could calculate the molecular weight between cross-links from mechanical models, so you could do mechanical testing, but because it's so related to swelling properties, you can also do swelling experiments to relate them. And by swelling experiments, I mean you just take the hydrogel, put it in PBS or water or some other solvent, and see how much water it takes up. And that's really related to the molecular weight between cross-links through the following derivation. So equilibrium swelling theory is what's used to derive this equation. It's kind of complicated. I'm not going to go through the entire derivation, but I'm going to just show how it works. So the Flory-Huggins theory was developed in 1953, and it just states that the degree of cross-linking or the degree of swelling of a cross-linked polymer is dependent on the elastic retractive force because of those cross-links in the polymer network. And it also depends on the interactions between the polymer and the solvent, which is water. And that makes sense too, that the amount of swelling, of course, depends on the interaction between the polymer and the water. You'll have more swelling if the interaction is stronger. So to go through the equations on how you might relate molecular weight between cross-links and swelling, Flory and Huggins started with the free energy of the system. And they said that this is simply the sum of the elastic retractive forces and of the free energy of the mixture of the polymer and the solvent, which is water. So the free energy, their change in free energy due to the elastic retractive forces can be determined based on rubber elasticity theory, which assumes that hydrogels are perfectly elastic for small deformations. And we'll go through that in just a little bit. But I want to start with how you derive the free energy for the actual swelling of the cross-linked polymer in water. And so that's the delta G of the mix. And that is based on the chemical potential of water inside and outside of the gel, which at equilibrium will be equal. So if you differentiate this free energy equation with respect to chemical potential, you get that the difference in chemical potential is the sum of the chemical potential due to the elastic retractive forces and to the polymer-water interactions. So in order to, so I guess that it's important to know that the chemical potential change at equilibrium is zero. So that means that you can make them the two sides equal to each other or to the negative of each other. So you can basically plug in zero for the chemical potential change and set the change in chemical potential of the elastic forces to the mixing forces. So we're going to go over those two terms. So the change in chemical potential due to the polymer-solvent interactions is based on this equation, which the derivation is out there, and you can look it up if you want, but I'm just going to go really quickly through that. And so it's based on the gas constant and on temperature. Of course, it's based on temperature because the energy that you add to the system is going to affect the interaction between the polymer and the solvent, and that, of course, is going to affect the change in free energy. And then V2S, you'll see a lot. That is the volume. That is the volume swelling of the swollen polymer. And so you have that. You can get that by dividing the volume of the polymer divided by the volume of the swollen gel, which is also known as 1 over Q, Q being the swelling ratio. And so the swelling ratio is the volume of the swollen gel divided by the volume of the polymer without being swollen. And so you can think of that as like the fold change of how much the volume changes when it swells in water. And that's often approximated as wet mass over dry mass. So if you swelled a hydrogel for 5 days and you assume it's at equilibrium at 5 days, you could just divide the mass of the swollen hydrogel divided by the one that you originally had, and that's Q, that's the swelling ratio. And so that can be really useful to tell you, how much that you can relate that to cross-linking density, and you can also relate it to diffusion within the gel. So that's a really useful property. Okay, so that is the equation for the change in chemical potential due to the mixing of the polymer and the solvent. So now we just need to get, we need to go into rubber elasticity theory in order to get that second term, which is where you would get the molecular weight between crosslinks. And then that's how we relate molecular weight between crosslinks to swelling. So that's why we're going through this. So the rubber elasticity theory assumes that the response of hydrogels to applied loads is approximately elastic under small deformations. Actually, this theory assumes that it's perfectly elastic under small deformations. So is that a legitimate assumption? This is data that I took a really long time ago of PBA hydrogels during my doctoral research. And so this is the stress-strain curve from 0 to 50%. And it is not linear, which means it's not elastic. However, if we look at small deformations, so less than 20%, and I cut off from 0 to 10% because there was noise there that would make it harder to see. But from 10 to 20%, they are linear. So that means that they are elastic under 20%. So it actually is a pretty good assumption to make in rubber elasticity theory. So as the name suggests, rubber elasticity theory was developed for rubbers and to describe the mechanical properties and the elasticity of rubbers. Without going into what this is, or how this was derived, you can see that the stress in the system is inversely proportional to the molecular weight between crosslinks of a rubber, which is a cross-linked polymer. It's just not really swollen with water. So to modify this for it being swollen with water, because that's what a hydrogel is, you just add the volume swelling ratio of the swollen polymer. So that's that term at the end there. So we can rearrange that a little bit and add in that volume swelling term, and you find that you can describe the molecular weight, you can describe the change in chemical potential of the elastic forces based on the molecular weight between crosslinks, and Mn there is the molecular weight of the polymer. And then you have the swelling of the polymer there. But this is all in terms of the elasticity theory. So this is not actually, so you have the swelling ratio, but it's just, it describes its mechanical properties. So if you combine those two equations that we had before, you had the one that was chemical potential of the mix and chemical potential of the elastic forces, and then you solve for molecular weight between crosslinks, this is how you can calculate it. So there's some terms here that I didn't go over yet. There's the molar volume of water, the volume of polymer in the relaxed state, the volume of polymer in the swollen state, we already talked about that. And chi is the Florey interaction parameter that describes, that's like a constant that describes the interaction of the polymer and the water. So this is a really ugly equation, but you can use it to just do simple swelling experiments and calculate the molecular weight between crosslinks. So I wanted to go through this because if you invent any new hydrogels, you're going to need to describe the molecular weight between crosslinks and the swelling properties, because it's very important for comparing these hydrogels. Well, first of all, for comparing it to other hydrogels, but also for predicting its properties in a variety of situations. So that's why we went through that. Okay, so rubber elasticity theory is one way to describe hydrogels, but you can also describe them with viscoelasticity theory. And that is because when you apply a load, like a compressive load, to a hydrogel, what is actually happening is the fluid is moving through the porous matrix. And there's different models that can describe it. I'm showing a model here which is called the biphasic theory, which is just what I just said. It's fluid flowing through a porous matrix. There's other things there's like a poro-viscoelastic theory. There's a whole lot of modeling that people have done to better describe the behavior. But suffice it to say that when you compress a hydrogel, fluid flows through the matrix. So that graph there is an example of creep testing of hydrogels in comparison to cartilage. So like I said, my doctoral research was on trying to make a biomimetic hydrogel that had the same properties as cartilage. Operative word being trying, because cartilage is a really unique material that has a lot of things that make its properties very different from just from a hydrogel that's made of one type of polymer. Hydrogels and cartilage are both pretty well described by the biphasic theory of fluid flowing through a porous matrix. And the equation up there is stress as a function of, or strain as a function of time. And there's some terms in there that are like the stress that you apply in a creep experiment. And you remember that creep is when you just hold a constant stress and you see how much the material creeps over time. And so that creep is sort of like how much fluid was moving around. And so the aggregate modulus is sort of like a measure of its stiffness in this sort of fluid flowing situation. So that's HA, that's how you can get that. There's an interaction parameter there, and there's time. So that's strain over time. And that's how and there's also permeability, which is K in that term there. So if you fit your creep data to a model like that, then you can actually calculate out aggregate modulus and permeability. And that's how you can, in a physiological test, you can compare a hydrogel that you made to something you're trying to mimic, like a tissue. So that's what I did. I compared a PVA hydrogel to cartilage. And what I found is that even though the water content of the hydrogel and cartilage were pretty similar in this experiment, their permeability to water was way different. So the permeability of cartilage was a lot lower than the PVA hydrogel, and that's because cartilage is not just a hydrogel of one polymer. It's got a lot of sulfated proteoglycans in there that have charges with a fixed charge density that attract water, which is polar. And so as water is flowing through the matrix, it doesn't flow as easily through cartilage as it does through a hydrogel. So that was the reason why this hydrogel would never behave the same as cartilage, because it didn't have that fixed charge density. And that's why cartilage is so stiff, because it can resist the mechanical forces, because that fluid can't flow as much as it would if it were just like a hydrogel. So because the fluid is an incompressible fluid, and because it can't flow, you have major resistance to applied forces. So that's a really interesting material that we have not been able to replicate in the lab. Like anyone has it, not just my lab. Okay, so I talked a little bit about PVA hydrogels earlier, prepared by freeze-thaw cycling and how that works. The reason why I wanted to use them for cartilage is because they have been extensively characterized and found to be really similar to cartilage in terms of a lot of these mechanical properties. Not permeability, which is what Chad just showed, but they do sort of, I think that there would be a way if you modified the backbone of the hydrogel chain to get that permeability to go down, and then maybe you could get it a lot closer to cartilage. But the problem with PVA hydrogels for cartilage is that when you implant it in cartilage, there's a complete lack of integration with the surrounding tissue. And so that image is of hydrogels that were implanted in human knees, and that image was taken at 3 months, and you can see there's just holes around the hydrogel. They're just sitting there in a hole, and there's no interaction. There's just a complete gap filled with fluid between the hydrogel and the cartilage. And so in my doctoral research, I basically showed that the reason why that happened is that cells cannot attach to PVA hydrogels, and so they'll never integrate because they can't migrate through or anything. So I found that the only way that you could get the cartilage to interact with the hydrogel is, one, if you added something that the cells could attach to, like a hydrophobic polymer. So that's why I added in a phase of a hydrophobic polymer that could absorb proteins that cells could attach to. And I also had to do a controlled release of proteins to actually have a chemoattracted concentration gradient that the cells would migrate towards. So if you just have the hydrogel, if I just had the PVA hydrogel, that would not have any interaction with the body. So I did some in vitro testing to see why didn't the cells attach to the hydrogels, or why didn't you have this? Why didn't you have that interaction? And this was actually just confirming what a lot of people have shown, that in this image you can see the porous hydrogels, and you have the hydrogels that were porous, but or they had large pores but no hydrophobic polymer, in this case PLGA. Those are the only cells that I found after seeding like 500,000 chondrocytes. But when I added in PLGA, the cells could attach, and then they could produce proteoglycans in cartilage tissue. So I confirmed what lots of people have found, that cell attachment will not happen on a hydrophilic polymer network. if you have, I guess there's some hydrogels that you can have cell attachment to. It depends on the polymer, but in general, if the polymer is very hydrophilic, the cells can't attach because proteins can't absorb. So that was really not good for cartilage, but it is pretty good for a lot of other applications like anti-biofilm or antibacterial surfaces, also called non-fouling surfaces. And that's because since proteins and cells, including bacterial cells, cannot attach to these materials, you can't have a bacterial colonization and you won't have infection. So they're also called stealth materials because the immune system doesn't really recognize them, but that's kind of an ambiguous statement. So we'll just stick with the fact that cells can't attach. It's really useful for something like urinary catheters, which has like 100% rate of infection if it's kept in there long enough because it's just really exposed to bacteria. So if bacteria can't attach, then it would be more resistant to infection. So coating something like a catheter with a hydrogel that cells can't attach to is very useful. The drug delivery applications are also really useful because if the immune system can't, if the cells of the immune system can't attach to to like a circulating drug delivery system like nanoparticles or microparticles, then they can't clear it from the system, and you'll actually have longer residence time of your drug delivery system before it's cleared from the system. So we'll go through that more in biomaterials too, but this is one application of the hydrogels. So I wanted to qualify all these statements by talking about fibrin gels, which cells actually can attach to and they like to. So this is a mixture of fibrinogen and thrombin. So when you mix solutions of fibrinogen and thrombin, the thrombin cleaves the fibrinogen and then makes it no longer soluble in the water that you dissolved it in, which causes it to precipitate out of the solution, but it's still swollen with the water, so it's a gel. It's a hydrogel. And it's a naturally adhesive hydrogel, and so it's marketed as Tissule by Baxter and also by TissueCall, or as TissueCall. And this is an image of it being used as an adhesive to attach a metal mesh for hernia repair to the tissue. So it's naturally adhesive. So that's fibrin. That's a really common biomaterial. Okay, so we talked a lot about proteins. Glycosaminoglycans like hyaluronan and chondroitin sulfate and heparin are really important parts of the extracellular matrix of connective tissues and of all tissues. But I was talking just a moment ago about how cartilage has those sulfated proteoglycans, and so this is a picture of, I think it's hyaluronan. And so you can see that it has the negatively charged sulfate groups on there. And that's what causes the resistance to flow that cartilage uses to resist applied forces. So all GAGs are negatively charged and attract cations, and that actually results in an osmotic imbalance that leads to swelling, which is what we've been talking about. So tissues actually swell like hydrogels do because of these GAGs. And so hyaluronan is really important for fluid film lubrication in cartilage of articulating joints because when you apply a force, the water will squeeze out, like we talked about in the creep experiment, and that actually forms like an articulating, a very lubricative layer. Okay, so hyaluronan is actually marketed as a supplement to I guess increase, they inject it into joints that are osteoarthritic in order to increase articulation and reduce the coefficient of friction. So a lot of patients report reduced pain this way by injecting that. But a lot of engineers and clinicians think that it's like a placebo effect because how long does it stay around? That's not really how hyaluronan works in the body. So, but I think it definitely works in the short term at least, and that it maybe puts in some extra cushion there. Okay, so alginate you guys are going to be working with in the labs. So it's derived from the cell walls of brown seaweed, and it's a linear copolymer of two different sugars, mannuronic acid and guluronic acid. The molecular weight can vary depending on processing, and it cross-links in the presence of divalent cations like we talked about earlier. So alginate is really useful for biomaterials and tissue engineering applications because it gels under such mild conditions that you don't really have to worry about it being having any residual cross-linkers that would be toxic to cells. and you can control the hydrogel properties based on a lot of different properties of alginate, like the concentration, the type of alginate, like how much glucuronic acid or mannuronic acid, and that's because of the bond rotation and that affects the stability there. And then based on the concentration of the ions, that'll change the crosslinking density, which will affect the properties that we discussed earlier. So alginate is commonly used as a wound dressing, and so it's used wound dressings in like burn wounds or for diabetic wounds or ulcers. They use hydrogels to keep the wound moist and provide a protective barrier against infection. So that's really just the physical properties of the hydrogels that make it useful there. Other than that, they're not really interacting with the skin at all. And then the other really cool thing about hydrogels is that they can be environmentally responsive. So the two most common types are ones that are responsible to temperature changes. So they'll be liquid at room temperature, and then solid or they form the hydrogel at body temperature. So that makes them injectable. So if they're liquid, they can go through a syringe, and as soon as they get into the body, they fill the shape of whatever they were injected into because they were a shape-filling liquid, and then they polymerized into a hydrogel. And so that's very useful. And pH-sensitive hydrogels might be collapsed at a low pH and then swell at a high pH. So that's extremely useful in drug delivery because you might want something to not release drug. If you take a drug like a pill, you don't want it to actually release the drug until it gets past the harsh environment of the stomach. It needs to get into the large intestine and the small intestine in order to actually release the drug into the bloodstream. So you want it to remain collapsed and not release the drug until you get past the stomach when the pH is low. So as soon as the pH increases, it swells and releases the drug. So we're going to go into a lot more about these smart hydrogels in Biomaterials 2 and 3. But I just wanted to mention them here because they're pretty cool. So in summary, natural polymers are nice because they give you the possibility to confer bioactivity into your biomaterial, and you can actually engineer interaction between cells and proteins of the body and your biomaterial, which is always going to be useful if that's something you can control. Hydrogels can be prepared from synthetic and natural polymers, and their properties can be varied widely depending on the crosslinking density and the network structure, but also just on processing and what sort of polymer you choose. And they have so many applications that they're really useful in the field of biomaterials. So that is natural polymers and hydrogels. Thanks.